Today we will start with the brain. So brain is the part of this central nervous system which lies in the center of the body. So brain is continuous with the spinal cord and it is a part of the nervous tissue, or it is made up of the neuron. So basic functional unit of the brain is the neuron. Or the nerve cell, that is the neuron. And brain is made up of grey matter and the white matter. So every part of the nervous system is made up of the grey matter and white matter. If I say grey matter, means I am talking about the cell body with the nucleus, with the cytoplasm and with the dendrites. If I say white matter, means I am talking about the axon or the nerve fiber with the nerve axon terminal, may or may not be covered by the myelin sheath. So that is the composition of the nervous tissue, that is the grey matter and white matter. And every part of the brain is made up of the grey matter and white matter. And we divide the brain into three parts: forebrain, midbrain, and hindbrain. And every part is made up of the grey and white matter, and brain is hollow from the inside. The cavity of the brain is filled with the CSF, which is a secretion from the choroid plexus or the tila choroidea. So the empty space in the brain is called ventricle. So every part of the brain is hollow for having the ventricle or the cavity. The cavity of the forebrain is called the lateral ventricle. Cavity of the midbrain is called the cerebral aqueduct, and the hindbrain, the cavity is called the fourth ventricle. Forebrain contains lateral ventricle plus third ventricle. So that is the what is called the brain. And brain is about 1.5 kg. Into weight. But we cannot feel the weight of the brain inside our skull because brain is floating in the CSF. Because CSF, which is produced inside the ventricular system of the brain, it's spread over the outer surface between the subarachnoid space, which makes the brain lighter and weightless. Brain and spinal cord, they are all covered by the three coverings that is called the meninges. The outermost is called the dura mater. The middle one is called the arachnoid mater. And the inside innermost is called the pia mater. So three coverings of the wrappings of the brain is there in order to protect the brain from the outer injury or from the head injury. And brain is well protected in the cranial cavity, which is made by the flat skull bones. So every measure has been taken to protect the brain inside the human body, because brain controls each and every part of our body. And if brain is damaged, a person or a patient is declared dead, we can revive the heart by injections, we can revive the lungs by ventilator or artificial respiration, but we cannot revive the brain. Why? Because of the neurons. Because neurons, they have lost the power of regeneration. These cells, they do not undergo mitosis. While all other tissues in the human body, they undergo mitosis. If they are damaged, the new ones are formed. But the number of the neurons which are present at birth, at the time of the death, they may decrease, but they never increase in number. So that is the specialty of the neuron. Now question arises, why they have lost the power of regeneration despite it has got the nucleus, which is the first stage of the cell division. Plus the neuron also have the centromeres. Even then, why it doesn't go into the cell division? The answer lies that they do not divide or they do not undergo the cell division or the mitosis, because the centromere, which is made for cell division in the neurons, it has got a function of protein synthesis. The most common and most special about the neuron is that the first step of the cell division. What is the first step of the cell division? That the liquid chromatin material of the nucleus will convert into solid chromosomes. And this step is absent in the neuron. So the nuclear material in the neuron nucleus remains liquid. It doesn't undergo the chromosomal formation, which is the first step of the mitosis, that is absent in this. This is the reason why neurons, they do not multiplicate. So brain is divided into three parts, and we will study the brain under these headings. First we will do the forebrain, then we will do the midbrain, then the hindbrain. Then we also use certain terminology for the nervous system. One is called the afferent. So afferent are the information or the impulses which are coming from the periphery to the brain or the central nervous system. And if some message is going away from the brain or the spinal cord, that is called the efferent. So E for exit, E for afferent, A for coming, that is called afferent. And these dendrites, they are mainly afferent in the function. While the axon terminal, it may go into afferent as well as the efferent. Then there are two more terminology we use. One is called the excitation, other is called the inhibition. Excitation is the response of the neuron at the target of the axon terminal. So when the message from the neurons goes into the axon, reaches the terminal, and there are the neurotransmitters are released here. About 50 neurotransmitters they have been found till date, but still the research is going on, like your periodic table. And when these released here, then the target structure, whether it is a muscle, maybe smooth muscle, maybe skeletal muscles, maybe the neuron soma or the body of the neuron, the response comes in the form of the contraction. That is called the excitatory action or the excitation. But if the impulse starting from the axon, starting from the neuron cell body, from gray matter and coming into the white matter, but if in between, if there is any blockade is there, or if any inhibition is there, then there will be no any response at the axon terminal. So such a impulse or such a process is called the inhibition action. So two things we are frequently using. Then we are also using the word relay, and we use this sign of the relay. As we do in the physics, in electronics, this is the sign we use, the positive and the negative. The current flows from positive. So, in the electric wires, electrons, they are moving. But in the neurons, the electrons, they are not moving. The salts or the sodium and potassium pumps, they are working, calcium, so ions, they are moving. So that is the difference. And whenever you touch any electric wire, you feel the current. But when the nerves are touched, or if the nerves, they are pressed, there is no current sensation, but there is with the touching, what happens? There will be pain. So pain is the most common modality of the nerve touching. So when you touch the naked nerve endings of the humans, he will or she will feel the pain. So that is the difference between the electric wires and the axons. So that is how we use them. This is a relay sign. This dot indicates gray matter or nucleus of the neuron or the cell body, and these two ends, they represent the axon terminal. So where the one neuron ends by the axon terminal, the message from the axon terminal will be shifted to the next neuron, that is the cell body, that is the nuclei. So we use the word gray matter as well as sometimes we use the word nuclei for that. And for the white matter, there are certain areas where there are bundles and bundles of the axons or the nerve fibers. For that, we use the word sometimes crus. Again, crus means a bundle. A bundle of 100 or the thousands of the axons. Then we use the word somewhere fasciculus. Again, fasciculus means bundles and bundles of axons or the nerve fiber. Then we also use sometimes peduncle. Again, the meaning of the peduncle is that again the bundle of the axons or the nerve fibers. Then we also use the word tracts. So tracts again means bundles and bundles of the axons or the nerve fibers. Depending upon the function of these neurons or the response of these cell bodies of the nervous system, we again use the word sensory and motor. Sensory and motor. So if I say sensory, remember sensory means S for S. If I say motor means M for M. What is the meaning of this? Sensory means any nerve or any part of the brain is labeled as sensory part, or a sensory neuron means, as far as means, it will start from skin or some special sense organ. So sensations, they are general sensations or special sensations. There are 4 general sensations and 4 special sensations. 4 general sensations, they are pain, temperature, pressure and touch. So our skin is sensitive from head to toe for these modalities of the general senses. Then the 4 special senses, they are hearing, smell, taste and vision. So 4 sensations, they are called the special sense organs. So from all these sense organs or from the skin, a neuron will start as a axon and it will end into some grey matter or into some nucleus. It will relate. So that means by the sensory area of the brain means sensations, they start from that. Then if I say motor, if any part of the brain is labeled as the motor or any neuron is labeled as the motor neurons means M for M, means the axon of that area of the brain, the axon of that neuron will end into some, M means muscle. It will end into some muscle. It may be the smooth muscle or it may be the skeletal muscle. That is the meaning of the M for M means M for muscle. And S for skin or the special sensory organ. So these are the small terms, but they are very, very important for the basics of the nervous system to understand, because nervous system is the most complex and difficult part of the human body. So in order to easily understand, you should know all these small things. Then it will be clear that what is the brain and how we will study the whole of the brain. Another thing about the brain is the applied anatomy. Now brain is affected by so many reasons or so many causes. But any part of the brain or the central nervous system is affected by 4 modalities, most common, and you have to remember this abbreviation T, T and H. T, T and H. First T stands for trauma. Our brain is affected by the head injury, roadside accident. If the skull is fractured or not fractured, even then the brain inside will be damaged. So trauma is the most common cause of the damage to the brain. Another is T, that is for the tumor. So tumor is a space occupying lesion. So small or large in size, any part of the brain is affected by a growth inside the brain or the cranial cavity. It will press some area of the brain and that area won't work, and patient come with the problem of that particular function of that area. So that is called the second T for the tumor is the space occupying lesion. So it acts as a pressure effect and the function of the brain is deteriorated. The tumor may be large size, may be small size. So that is another modality due to which the function of the brain is, or any part of the nervous system is deranged. Then third T stands for thromboembolism. Thrombus, like coronary arteries, the cerebral arteries, they are also end arteries. They do not have what the collaterals. They are the selfish arteries, they only supply their own area. They won't anastomose. And this is the problem. It leads to the obstruction in the blood flow, or the arterial flow of the any part of the brain can be affected just by the formation of the thrombus or the clot. The reason of the thrombus may be so many. So any supply to the area of the brain is affected when the any capillary or the any artery is obstructed by some atherosclerosis or some other parts because of their end artery nature, so that area go out of the order. So thromboembolism is another vascular cause of the affection of the brain. Then H stands for hemorrhage. So bleeding or the hemorrhage is the another cause of the affection of the brain, most common, and it is very common in the patients of the hypertension. So high blood pressure may lead to the rupture of the any artery supplying the any part of the brain, and rupture of the artery lead to the bleeding inside the brain, and that blood is dissolved in the CSF. As we know, the blood vessels, they are bathing in the CSF. And brain is very soft. If you touch the live brain during craniotomy, the brain is very soft. How much soft? Not like this jelly-like soft, but it is like the dough we make for chapatis. It is so soft. So as the blood vessels, they are lying in the subarachnoid space and bathing in the CSF, the rupture of any vessel lead to the bleeding, and that blood will be dissolved in the CSF. It lead to the increase in the volume. And when the volume is increased, pressure is increased. So that lead to the intracranial pressure increase. And intracranial pressure lead to the derangement of the brain functions. So that is the problematic. If that bleeding is on the surface of the brain, then it lead to the rupture and bleeding, and that blood is clotted. So this is the nature of the blood. When the blood will come out of some blood vessel, it undergo clotting. All clotting factors, they accumulate there. The fibrin, fibrinogen and everything, you know, the Christmas factor and so many factors. So all these factors, they localize at that area. That lead to the formation of the clot, and that clotted blood at the site of the, maybe inside the grey matter, maybe outside the grey matter, or inside the white matter, maybe there. So that blood is clotted, and that clotted blood is called hematoma. So he means blood, toma means like a tumor. So that clotted blood is called hematoma, and it acts like the same as the damage is caused by a tumor. And this clot had the power of shrinkage also. So with the subsequent passage of the time, this clot may decrease in size and patient may be relieved of the symptoms of the brain damage. It depends upon the size of the hematoma. If the size is bigger, some deficit, neurological deficit will remain for the lifelong. But if this is smaller in size, then we say to the patient that it may be resolved with the duration of the time, and your neurological disorders will be okay, and this will be corrected with the passage of the time. So that depends upon the CT scan report and MRI, that what is the size of the hematoma and what is the site of the hematoma. That is very, very important. So that is the general term we use before we starting with the more meticulous and more deep study of the each and every part of the brain. Then we have to do. These terms should be on your fingertips. Then we take one by one. As we know, every part of the brain is having different function to perform on the human body, and brain controls the human body. That is the universal truth. Even a rickshaw puller knows that our brain controls the human body. Plus, he also knows that right side of our brain controls the left side of the body. So these are the general terms which are present in our society, and these two things, everybody knows about the brain. Now we will go into the depth of the knowledge of the what is the brain and how it functions, and why different parts of the brain controls different part of the human body. This should be your quest. It should be your eagerness, that you must be eager to know that which part of the brain is controlling which part of my body. So then only you can understand what is the brain and what are the functions of it. So first of all, we will take the forebrain. If we take the forebrain, we know the forebrain consists of two parts. One is called the cerebrum, and another is called diencephalon. So we will take one by one. First we will take the cerebrum, then the diencephalon. Now cerebrum is the largest part of the brain. If we see the surface area of the volume, then we will see that cerebrum is the largest part of the brain, and as every part of the brain is hollow from inside, so it is also hollow. and filled with the CSF. And every part of the brain is made up of grey and white matter, so it is also made up of the grey and white matter. Plus, in some of the part of the brain, grey matter is inside and white matter is outside, or vice versa. In the cerebrum, the grey matter is outside as well as inside, but white matter is inside. Grey matter is of both the sides, but the white matter is inside the cerebrum. And the cerebrum, we remove the brain from the skull cadavers and we study it and we put it in the tray and we examine what is called the brain or the cerebrum part of it. So when we study the cerebrum is like my hand. There are two cerebral hemispheres, right and left. And if you join them, they are almost oblong in shape. It is not circular, it is not oval, it is oblong or ovoid in shape. And if I touch the upper surface, this is called the superior surface of the cerebrum. If I touch here, this is the lateral surface of the cerebrum. And below, this is the inferior surface of the cerebrum. And in the midline is the medial surface. And two cerebral hemispheres, they are not glued together, but they are separated a little bit by a longitudinal fissure in which lies the sickle-shaped falx cerebri, which is the derivative of the tough, durable durameter. So durameter separates the two cerebral hemispheres' medial surface. While the two cerebral hemispheres, they are also joined together, not by the grey matter, but they are grey matter of the right and left cerebral hemisphere is joined by the horizontally running white matter bundles that is called the corpus callosum. So that is how we use the terminology for the cerebrum. If we see on the surface of the cerebrum, what we see that is scattered is the grey matter. Outside is the grey matter means cell body with nucleus, with cytoplasm, with dendrites. So they are forming the outermost core or outermost covering of the cerebrum, that is called the, like kidney, sometimes it is also called cerebral cortex. As kidney has got outer periphery part is called cortex and middle part is called the medulla. So sometimes we also use the word for the grey matter of the cerebrum, which is very surface area, is very large on the surface. It's called the cerebral cortex. With the duration of the time, with the aging process, the neurons of the cerebral cortex, they diminish inside because they are damaged with the senile changes. So after the 60 years of the age, average, the neurons they damage, and these are called the senile atrophy. That's why after 60 years, there starts the problems of the brain, that the loss of memory, loss of reflexes, loss of so many things they undergo loss after the age of 60, whether it's a male or the female. So this is only anatomical basis is that the neurons they undergo damage or atrophy. And the thickness of this cerebral cortex or the grey matter also decrease in the thickness. When you see the MRI and the CT reports after the age of the 60, there is generalized atrophy of the cerebral cortex. And it is attributed to the simple senile changes. Diffuse cerebral atrophy findings in the CT scan after the age of 60 or so is called the diffuse cerebral atrophy. So that is how we do the cerebrum. That is the largest part of the brain. Another grey matter is present inside also. The inside grey matter, which is present inside the cerebrum, part of the grey matter of the cerebrum is very much of, it is made up of the compact bundles of the grey matter. Thick bundles or thick group of the neurons or the grey matter, which are very compact inside, and they are called basal ganglia. So basal ganglia, they are also part of the grey matter of the cerebrum. Then there are certain areas which are hidden from our view of the superficial surface, and that is called the insula also. So the cerebral grey matter, when we talk about cerebrum, we divide into study the grey matter and white matter. So first we will study the grey matter, then we will switch on to the white matter of the cerebrum. So if I say the grey matter of the cerebrum, means I will talk about the cerebral cortex, which is the peripheral part. Then I will talk about the insula, that is a little bit buried part of the grey matter of the cerebrum. Then I will talk about the third part, that is called the basal ganglia. That will do one by one, that what are these parts, and then we study the cerebral. Then we do the next time. Then we also use the word that is called the brainstem. So brainstem is the word used for the three parts of the brain, that is the midbrain, bones, and medulla oblongata. As we know, the brain is continuous with the spinal cord, covered by the meninges lying in the cranial cavity made by the skull bones, and the brain ends at the upper border of the atlas, that is the C1. Upper border. And so here the brain ends. The lowest part of the brain is the medulla oblongata at the upper part of the C1 or the atlas. And this is the same point from where the 45 centimeter long spinal cord starts. So that is the CNS. Part of the CNS is the brain and the spinal cord. As brain is attached with the 12 pairs of the cranial nerves, spinal cord is attached with the 31 pairs of the spinal nerves. While the cranial nerves, some are motor, some are sensory, some are mixed. But all 31 pairs of the spinal nerves, they are mixed, because they contain both motor as well as the sensory fibers. So as the cerebrum is the largest part of the brain, as the forebrain consists of cerebrum and diencephalon, and the midbrain is the smallest part of the brain. It is 1 inch long and 1 inch broad. It connects the forebrain with the hindbrain. While the hindbrain consists of three parts, that is the bones, medulla oblongata, and cerebellum. So the midbrain, bones, and medulla, they are called the brainstem. Why it is called brainstem? Because brain looks like a tree. As we know, tree is supported by the stem. Same is the appearance of the brain. So this is the part of the tree. So this is the part of the brain that is largest part of the cerebrum, and it is supported by obliquely placed midbrain, bones, and medulla oblongata. So it resembles the tree. So as the cerebrum is supported by the midbrain, bones, and medulla, so it looks like the appearance of a tree. That's why we use the word brain stem, like the stem of the tree. That is the midbrain, bones, and medulla oblongata. So this is how we explain the parts of the brain and their structure. That's all.